Hello. Can everybody? All right. Can everybody hear me all right? Awesome. Cool. If you can like pull in other people from outside, go ahead. It's a big room. And get as close as you feel comfortable. Okay, thanks so much for coming to this session and for this talk in particular, and to Deepa for organizing and inviting us all here to review adaptive genetic variation. And I'd like to acknowledge my co-authors on this work, my grad student, Chenga Patek, as well as Lapo Frati and Reed Brennan, two other co-authors who helped think through some of the analyses that I'll share with you. Taking careful notes in a notebook, observations, that's how naturalists for centuries have documented differences among populations. And these have been associated with environmental changes. And then experimental biologists made things a bit more fun and satisfying by doing things like reciprocal transplanting to test for local adaptation, or moving things together into a common garden environment to reveal the genetic bases or the physiological bases of genetically controlled differences. And over the last 60 years, things have really taken off in terms of molecular and statistical approaches that allow us to really dig down deep, get into the hood to figure out the genetic and physiological bases of these adaptive differences and traits. But we're at an exciting time right now where this classic model that a phenotype is the result of a genotype in the context of its environment is really bursting at the seams. So now we can link genomes to phenomes, linking complex polygenic traits to their genetic bases, and we can do this in natural populations and in emerging model organisms. We also have this devastating experiment going on where the environment in which we live is changing at a geologically unprecedented rate. So we can study if and how organisms might adapt to this rapid environmental change. And the relationship between genotype, phenotype, and the environment is actually a bit more complicated than we thought, where there's a potential role for the microorganisms that live in and on an organism to affect that phenotype. And the epigenetic variation can be controlled or affected by the environment and the genotype to affect the phenotype. And lastly, we can really make this genotype to phenotype link in ways that were never before possible, using things like CRISPR-Cas9 to make the connection. But how do we get to these frontiers? How do we study adaptive genetic variation? A number of studies use ecological genomics or landscape genomics, where you can associate genetic variation with environmental variation. Or instead of space, you can study things through time using experimental evolution, for example, as we did with these marine copepods that we experimentally evolved in high temperature and high CO2 conditions for 20 generations, and then reciprocally swapped them to see what the consequences were of long-term adaptation. Or you can get lucky with a before and after sampling in time, like Bumpus did with his house sparrows, or Arnold and Landy and Arnold with the beetles, or before and after a drought for the Grant's finches, or a cold snap for Campbell Stanton's Anolis lizards, or before and after a disease event for Pierre de Witt's abalone. And a number of other studies have pointed out the importance of regulatory variation in local adaptation, such as desert adaptation in the house mouse, or inland versus coastal adaptation in the monkeyflower, the yellow monkeyflower mimulus, or marine versus freshwater stickleback fishes in adapting to those local environments. But what if there is spatial variation in the degree of temporal variability? How about local adaptation to environmental variability? This has been studied to a much lesser degree, which there's an excellent system in which to investigate this, which is the California current ecosystem. And what we're looking at here is along the west coast from Washington down to Southern California here, and you can see the degree of heterogeneity in pH conditions, with more variable sites located in the northern part of the coastal range. So you have both this persistent spatial differences in temporal variability with more heterogeneity in the north, which is driven by this natural process called upwelling, where low pH waters are brought up from the deep ocean. And there's an organism that has evolved in this environment called the purple sea urchin, Strongylocentrotus purpuratus, that lives from the cold waters of Alaska to the warmer waters of Baja California, Mexico. You can find them in rocky inter- and subtidal reefs, where there are both males and females, and they broadcast spawn, releasing their eggs and sperm into the water column where fertilization occurs. And they create these beautiful little larvae, as you can see here, with these long skeletal arms that they use for feeding. So it's an important trait for survival. These arms are made out of calcium carbonate, so they're sensitive to pH. But these larvae can spend weeks to months in the water column before they recruit to a suitable place to live, which is a lot of dispersal potential. So all of the previous work in this species has shown no population structure in neutral loci due to this high dispersal potential. They also, because of this broadcast spawning, they have large transparent eggs, which has made them a model for developmental biology for over a century. So there's extensive genomic and developmental genetic resources that make this an excellent organism to study. But they're not impervious to ocean acidification, or the other CO2 problem, as it's called. About a third of the CO2 that we emit into the atmosphere is absorbed by the Earth's oceans very kindly. But that's not without consequences, so 22 million tons a day. And as the CO2 dissolves into the ocean and reacts with water, it consumes carbonate ions, reducing the availability of those ions for calcifying organisms, and also lowering the pH, so it's a double whammy. In the last 100 years, the pH has dropped from pH of 8.2 to 8.1, which might not seem like much, but recall that the pH scale is a log scale like the Richter scale, so this is about a 25% increase in acidity, which of course poses a challenge for the growth and maintenance of skeletons and shells, as you can see in this beautiful sea butterfly here as its shell dissolves as it spends some time in low pH waters. And there's evidence of adaptive capacity to low pH conditions in the purple sea urchin. From our previous work, one study, which was a common garden acclimation study of purple sea urchins of adult urchins for 3 years, we found that 99.6% of the genes were expressed exactly the same, except for a suite of biomineralization genes, where some of them were more highly expressed in the southern urchins compared to the northern urchins. And this also corresponded with a higher ability to regrow spines. And in a number of other studies using larval selection experiments, we've shown that there's standing genetic variation for being able to evolve or at least survive low pH conditions. And in the idea of new mutations versus old mutations in the Brennan et al. Study, we showed that the variation that was important for being able to survive extreme low pH conditions were rare variants, whereas the everyday kind of pH fluctuations that you experience were being survived by balanced polymorphisms, at least polymorphisms that were at higher frequencies in the natural populations. So there's evidence that there's adaptive capacity, but we have very limited information on regulatory variation from this species. Which leads me to my graduate student, Chenga Patek, who wanted to address this question. Can we detect signatures of adaptation, local adaptation to pH variability in the purple sea urchin? And to specifically test the hypothesis, if there are signatures of adaptation to pH variability, that they will be in the regulatory regions of the genome. To address this hypothesis, she chose sampling localities based on the degree of pH variability. So using the same data from that Chan et al. Paper, she selected 7 sites. 3 of those sites, noted with the red stars and the circles on the map, had really high variability, and with pH that's dropping down below levels of pH 7.8 at a much more frequent time scale. She sampled 20 individuals per population for a total of 140 whole genomes sequenced across all of these urchins, and did paired-end long-read sequencing using the NovaSeq platform, which resulted in about 6x coverage across the genomes of all 140 individuals and had a mapping rate of about 81.6% to the latest reference genome of the purple urchin. She then calculated genotype likelihoods and to then identify polymorphisms, finding about 16 million polymorphisms that she then filtered down to about a set of 1 million high-quality single nucleotide polymorphisms. And for the purposes of this study, we're just focusing on these SNPs, though we've got ideas for looking at indels and inversions. And then calculated FST from these, as well as a number of other population genetic metrics. So looking at all of the loci, all 1 million polymorphic SNPs across, or all 1 million SNPs across the 140 individuals, you can see that there's no population structure. So each dot here is an individual colored by the population where they were sampled from. And this represents basically a blank slate for tests for selection. And there's incredibly high genetic diversity, pi of 0.009, or about 1%, which is on the order of what you see for many marine invertebrates, but orders of magnitude higher than what we see for mammals, for example. And we see, again, no population structure, no signatures of isolation by distance. Any way we look at the data, there's no structure. So despite this, looking at the genetic, or testing for genetic differences using FST between the high versus the low variability populations, we found a strong signature of about 1,000, or 10,000 SNPs that had differentiated between these sets of populations. So here we're looking at a Manhattan plot across all the chromosomes, and you can see here's the distribution shown on the left and these outliers here. And it looks like they're randomly distributed, but if we use functional enrichment analyses to ask what are the types of genes where we're finding these polymorphisms, we can see that they're concentrated in genes related to cell signaling, ion transport, and cell adhesion, all functional classes of genes we've found before to be important for surviving low pH conditions in these sea urchins. So where are these genes in the genome, to get at Chenga's original question? So if we classified all these polymorphisms based on the functional or the genomic regions where they are, so in introns, non-coding regions, exons, 5', 3' UTR, enhancer regions, or long non-coding RNAs, we can find that there's a dramatic overrepresentation of these pH-associated SNPs in the enhancer and long non-coding regions of the genome. In fact, all of these are significantly different than expected. What you're looking at here is the number of observed SNPs over expected based on the genome-wide distribution. So a value greater than 1 is showing more observed than expected, and a value less than 1 is showing fewer observed than expected. So all of these are significantly different than expected, except for this 5' UTR. So we've got also an underrepresentation of intronic SNPs in these pH-associated, or pH variability associated SNPs. So there's a strong signature of low pH adaptive SNPs enriched in regulatory regions, but of course there's also signatures of important exonic SNPs to follow up on as well. And being a systems or network biologist that Chenga is, she wanted to ask what are the relative positions of these SNPs in interacting genes across the genome. So she built a network of all the protein-protein interactions and asked the number of neighbors that these pH-associated SNPs had. So looking at, these are violin distribution plots. So the non-outlier genes here's the distribution, and then those in gene bodies, the pH-associated SNPs in gene bodies versus the pH-associated SNPs in promoter regions. So you can see that they are underrepresented in promoter regions, suggesting that These putative adaptive regulatory variants are more peripheral, so they're not central in these protein-protein interaction networks. They're less connected. Similarly, we wanted to ask when these genes are expressed during development. Since we know so much about development in the purple sea urchin, we have a suite of timing of gene expression data from 72 hours, 48 hours, and 24 hours. And in a similar plot as what I was showing you before, where we were looking at observed over expected, we can see that in the gene body region, we have a dramatic overrepresentation of these early developmental genes, of SNPs occurring in these early developmental genes. But when we look at the SNPs in promoter regions, they are dramatically underrepresented, which I think is a really cool result, showing that this is only for SNPs in gene bodies, and that we see the opposite pattern for the SNPs in promoter regions, suggesting, perhaps not unexpectedly, a conservation of these early developmental gene regulation. So local adaptation doesn't mess with the canalization of development. And I mentioned to you this trait before of biomineralization genes, so the ability to be able to grow these beautiful skeletons as you see here. So if we plot all the SNPs and make a phylogenetic tree with all the SNPs and color code the populations that we've sampled in red here at the end of this, based on least variable in light white colors and more variable in more red colors, we can see that there's no real relationship between the clustering of the populations and the degree of pH variability. However, when we make this phylogenetic tree with just the SNPs that are in the biomineralization genes, we can see that now there is a clustering, of this locations based on the degree of pH variability experienced in nature, just for these biomineralization genes, which is super cool. And if we want to zoom in, this is about 77, the SNPs in about 77 genes. If we zoom in on 4 of these genes here, we have what we're looking at here is the minor allele frequency of 4 of these genes plotted along with a metric for the pH variability across all 7 populations organized from south to north here, with the pH variability in black. The blue and the red are both SNPs in promoter regions of these 2 genes in this extracellular matrix protein 3 in blue, and then in red, the spicule matrix protein 30. And then 2 exonic non-synonymous mutations in orange and green here in this mesenchyme-specific glycoprotein, SNP30, and in Notch, this really critical cell differentiation gene in green. And if we look at the relationship between the high FST region, or the high pH variability regions here and the low pH variability regions here for this single Notch gene, that's a 20% difference in allele frequency, which is a huge deal for a sea urchin if you've ever looked at genetic variation in a sea urchin. You did see the PCA in the beginning, so you can see why I'm excited about that. Okay, and just to zoom in a little bit more around this Notch gene, we're looking at that non-synonymous mutation here in this exonic region and a couple of intronic SNPs here. The blue dots are showing the FST of other SNPs in the area. This is the gene body of the Notch gene. And you can see, looking at the green line, which is showing the pairwise nucleotide diversity, that there's a slight reduction in nucleotide diversity around this Notch gene, so suggesting that maybe there's some sweeping action happening here, which again is huge for a sea urchin that has an incredible amount of genetic diversity and gene flow. So that would be an exciting candidate to start with, but the one that Chenga is starting with, she's passionate about regulatory variation, is this EMC3 gene, where she's got a couple polymorphisms, 2 SNPs in the promoter region. So she's developed, or she's using a GFP reporter assay where she's going to study the effects of these mutations on the expression of this green fluorescent protein. So this is a picture of one of her injected embryos. but you'll have to stay tuned for the results on that. So to just summarize what I've showed you, that there's no global population structure in these purple urchins. And despite this lack of population structure, we do see signatures of low pH associated or pH variability associated SNPs in regulatory regions of the genome. But those are further not random. So they're peripheral in networks, they're not central, and they're not expressed in genes or in the promoter regions of genes expressed early in development. Biomineralization genes, again, seem key to this signature of local adaptation, and we have a number of excellent candidates for future study. But the big take-home messages are that there is adaptive capacity and signatures of adaptation to pH variability despite the extremely high gene flow in these organisms, and that gene regulation will be or seems to be important for adapting to variable conditions, particularly pH variability. And lastly, that this work combined with work from my lab in the past suggests that there is standing genetic variation that will be important for adapting to future global change conditions. But there, of course, are costs. There can be depletion of this genetic variation, which, again, relates on the ability to replenish that variation. With that, I'd like to thank you all for coming to this talk. All the code for the work presented can be found on our lab GitHub page. I'll be hiring 2 postdocs, one to help with this and work on urchin functional genetics, and another to work on our copepod experimental evolution work. Of course, thanks to all of my lab for being such an amazing community of people to work with and play with, and to the amazing crew of people who collected urchins for us at the beginning of the pandemic. So Pete Ramundi, Bruce Mengi, Sarah Gravum, Pete Halme, and the entire PISCO team, and the National Science Foundation for funding this work. And thank you so much. I'll take any questions. Yes. How long lived? The dispersal range? Yeah, so it's estimated from other invertebrates that it can be tens to hundreds of kilometers. So it's hard to tag them with them being so small, but with other people have done studies with genetics and doing sampling of plankton toes to try to estimate, the dispersal capacity of a variety of marine invertebrates. And that seems to be, you could get stuck locally or you could travel hundreds of kilometers. Yeah. Question. Yeah, Ed. Scott.
How key was it to find out whether a patient SNP was like in a promoter of the genome
Yeah, that's a great question. We actually had, so there was a lot behind the scenes that Chenga did with this just because she's so interested in this regulatory variation. So with sea urchins, being so much being known, the enhancers, like what we call as enhancer, a lot of those are experimentally validated. So she's really excited about those 77 enhancers that she's found, or SNPs and 77 enhancers. What we call promoter, I think, is more bioinformatically determined. So there's a degree of confidence depending on the sites. But because there's been so much with ATAC-seq and other sorts of validation techniques, we're a little bit more confident in some of these. So I feel much more confident about these results than I would if I were telling you about copepod enhancer regions, just because we know much less about those. But yeah. Yes.
We focused on, of course, the pH, but do you have anything to tell us about the thermal data?
Yeah. Yeah, that's a great question. And that'll definitely be, that's something that Chenga is working on right now. And that'll be another study coming out of this rich data set. So yeah, she's got probably another 10 different environmental variables that she's looking at that, of course you can in her first pass analyses, really one of the strongest, or probably the strongest signature was to this pH variability. But of course, it's also confounded with just, amount of time spent at pH less than 7.8. So it could be the variability, or it could be just the extreme low pH. It's really, it's correlative, but there's definitely interesting signatures there related to temperature too. Yeah. Good question. Yeah, Lauren.
Are they variable in the same way, or are there different time scales of variability? I guess you just said that it might just sometimes be minimal pH, but can you figure anything out about the temporal dynamics based on their genetics?
Yeah, that's a great question. It's so when we're talking about these larvae, they're in the water column for the weeks to months. So what they're experiencing, I think, these pH data were collected near shore, so they might be slightly offshore. But then the sea urchins as adults are so long-lived. You can pick up an urchin and it could be 50 years old. So that's just kind of crazy. So the time scale on which this pH variability is acting really depends on the life stage of the organism and whether they're out to sea or trying to recruit to the rock. But the variability that is experienced in nature is on the order of within a day. You get cycling variation in the very near shore in the intertidal and subtidal. But then, of course, there's longer term patterns as well. So it's a tricky thing to get the environmental variation and the organismal life history to line up to know what exactly they're experiencing and on what time scales. But it's an important question. Yeah. All right. Thank you so much.